This is the last talk of this session is by Tang. He's my graduate student, has been in my lab for the last about 4 years now. He came from aerospace engineering, so I thought that what can he do in biology, but he started to learn cell culture, and then he got interested in cancer. He started working with Professor Kuhlenschmidt, who is in pathobiology, and then he started to look at what happens when cancer cells go to metastatic stages. The initial idea was to find out whether the cell-cell adhesion decreases as metastasis happens, but then he figured out that there is much more interesting things happening beyond just cell-cell adhesion. So that's what he's going to talk to you today.
Thank you. Okay, good afternoon, everyone. My name is Tang, and today my talk is the mechanosensitivity and cancer metastasis. First of all, I would like to thank my advisor, Professor Taher Saif, and Professor Kuhlenschmidt. And this work is in collaboration with Dr. Teresa Kuhlenschmidt, Dr. Josh Zhou, Mr. Philip Bill, and Professor Philip Wan. First of all, we know 90% of the cancer deaths are due to the cancer metastasis. During the cancer metastasis, the malignant cancer cells firstly make the decision to detach from the primary tumor. And then those cells down-regulate their cell-cell adhesion and upregulate some cancer invasive proteinase to invade into the circulation system. Driven by the flowing blood, those malignant cancer cells travel through the human body and reach various distant organs. Then those cancer cells escape from the blood vessels to settle down in those distant organs. Then they generate numerous secondary tumors. It is those numerous secondary tumors kill the patient, not the primary tumor. If the primary tumor is detected on time, they can be cut and the patient will cure. But if there are numerous secondary tumors generated, there are just too many tumors to be cut. The problem to cure the cancer is mainly because we don't know which signal causes the malignant cancer cells to make this decision to detach from the primary tumor at the first place. So in our work, we discovered that by only changing the mechanical environment alone, the cancer cells can make the decision to become more malignant and to prefer some metastasis-like phenotype and behaviors. The experimental systems are as follows. We used the substrate with variable stiffness to culture the cancer cells. Those substrate stiffness are varied from 1 kPa as softer as human brains to intermediate soft 20 kPa as softer as the human muscles to very stiff substrate like 3 gigapascal polystyrene substrate. This is conventionally used for the cell culture. We choose the human colon cancer cell line HCT8 as our model. The colon cancer cells HCT8 is obtained from the primary tumor of the human patient, but not metastasized yet. It is a cell line. We use the normal cell, MA104, and bovine endothelial cells as the control. To make the identical chemical environment on the gel surface, we use the fibronectin, laminin, or collagen as the extracellular matrix to enhance the cell adhesion. We only change the stiffness of the substrate, mechanical stiffness of the substrate. This is what we observed for the normal cells, for the MA104 and the bovine endothelial cells. When the normal cells are cultured on the soft substrate, like a 20 kPa substrate, they form the clusters. They left some blank area behind them and form the clusters to form the cell-cell junctions. When they are cultured on the harder substrate, like polystyrene substrate, they, as you guys already know, they form the monolayer. And due to the contact inhibition of the normal cells, they stop proliferate until the confluence is reached. We did the same thing for the cancer cells. We find on the soft substrate, they also form the monolayer, form the clusters. On the hard substrate, they form the monolayer. At the first look, they behave the same, normal cells and cancer cells. However, if we keep looking longer, something interesting happened. Constantly, we observed on the 7th day of culture, there are some single cells detached from the edge of the cell colony and migrate away. This detachment keeps going until after 10 to 14th day, the whole colony falling apart into numerous single cells. And those cells keep proliferating. In contrast, on the very stiff polystyrene substrate, the cells keep in the monolayer. And since they are cancer cells, they don't have contact inhibition. They start to pile up on the monolayer. But they never show this dissociation phenotype. So when we observe this phenotype first time, our first question is those really the cells? Are they just dying? So we did the live dead assay to test the viability of those cells. We found those dissociated cells actually are living even better than the cells in the colony. Those green color indicated alive, and red indicated dead. Not only alive, those cells, dissociated cells, are also mortal and metastatic. Here we show the migration trajectory of those dissociated cells. They migrate away from the cell island, cell colony, with the maximum speed 1 to 2 micron per minute. Here I show the time-lapse video recording the dissociation process of a small cell cluster. So you can see within 7 to 8 hours, the cells are dissociated completely from the cell cluster. This picture shows a zoomed-out view of the dissociated cells on the whole gel. The diameter of this gel is about 1 centimeter. So here you see this is after 14 days. I'm sorry, the top of the caption is cut off, but this is after 14 days culture of HCT8 cells on the 20 kPa gel. So remember, we start up with the epithelial phenotype cells, but now we end up with all the rounded shaped single cells. From now on, we call those epithelial type phenotype cell E cells, and the rounded dissociated cells R cells to make it simpler. Then we harvest those R cells as well as E cells to perform the invasion assay. What we use is the basement invasion assay. We cultured those E cells and R cells on the top of a 100 micro thicker basement membrane. We culture the cells with the media without any serum and put the media with serum beneath the basement membrane. So due to the camera tracking, those cells will penetrate the basement membrane. After 2 days, we find at the bottom of the basement membrane, the R cells penetrate 15 times more than E cells. These surprising results told us this cell dissociation process is not a simple cell dispersion. It's actually a cell state transition. After triggered by the appropriate mechanical microenvironment, the cells actually transferred to a more malignant cell state. Then, next question we ask is, why those dissociated cells don't form the cell cluster again on the soft substrate? Another parallel question is, if these cells can be driven to dissociate due to the appropriate mechanical signal, can they be reverted back if we change the mechanical signal back to the unfavorable signal, such as the polystyrene substrate? These two questions can be answered by one experiment, which is to harvest the dissociated cells from the polyacrylamide gel and replate them back to the fresh polyacrylamide gel as well as the fresh polystyrene substrate, soft and hard substrate. What we observed is, very surprisingly, after those dissociated cells are replated back to the fresh, soft, and hard substrate, they behave identically. If I don't tell you what substrate they are on, you will not tell whether they are on the soft or hard substrate. Totally opposite to the original cancer cells' behavior. On the soft substrate, they form the colony on the hard substrate, they form the monolayer. Those dissociated cells totally remain dissociated, remain rounded phenotype, regardless of the substrate stiffness. This experiment told us two things. One is the cell transition. This cell state transition is stable and permanent. It can be reverted. It can be triggered by the mechanical signal, but it cannot be reverted by the mechanical signal alone. Second conclusion we find from this experiment is these cells actually lost the substrate stiffness sensitivity, which means they are more automotive, and that's an important hallmark of the cancer metastasis. As I said at the beginning, during the metastasis, especially at the onset of the metastasis, cancer cells need to downregulate their cell-cell junction, to downregulate the cell-cell adhesion molecules to fill the cell-cell junctions. So to test whether the dissociated cells also behave in the same way, we stand the E-cadherin on both E cells and R cells. E-cadherin is a well-known cell-cell adhesion molecules. Here, the green indicates the E-cadherin expression levels. On the top two pictures showing the E-cadherin expression level in the cell colony, and the second two pictures showing the E-cadherin expression in the dissociated cells. By evaluating the fluorescent intensity, they are downregulated 5 to 6 times. We also verified this E-cadherin expression level using the Western blotting. They are really downregulated. Also, as the dissociated cells are replated back to the polystyrene substrate, even the mechanical microenvironment is changed, they don't upregulate the E-cadherin. They don't restore that surface cell-cell adhesion molecules. So here, this experiment verified the downregulation of the cell-cell molecules. It only tells us the presence of the cell-cell adhesion molecule, but does not tell us the function of the cell-cell adhesion. So that's not sufficient. So to test the function of the cell-cell adhesion, whether that is really a field, we use the cold counter assay in Professor Kuhlenschmidt's lab. In this experiment, what we did is we firstly suspended the cells, the R cells, E cells, and the normal cells as control. When cells are suspended, we put them into the identical series of vials and let them settle down in the 37 degrees C water chamber for varied time, from up to 2 hours. As the incubating time increases, cells tend to aggregate. In the cold counter machine, there is a small hole, which is small enough, just small enough to let only one cell to get through. So at a different time point, we pick up the cell samples to let the cells penetrate the holes. More cells penetrate through the holes means more single cells remain in the tube, in the vials, which means cells are less tending to form the cell-cell adhesion. So our data suggests throughout the 2 hours incubation, the R cells remain constant. You may notice the single cell amount of R cells mostly constant. For the E cells, they reduce, and especially for the control, they reduce dramatically, meaning the cell adhesion activity is going on. Then I also developed a traction force microscopy to study the interaction between the cells and the substrate. The traction microscopy is used to measure how much traction force cells can exert on the substrate underneath them. What I find is, for the E cells colony, they apply maximum 300 Pascal traction force on the soft substrate. In contrast, after the cell dissociates from the colony, they apply very weak traction force on the substrate. The maximum is 100 to 150 Pascal, 2 to 3 times level down. This experiment tells us the cells not only reduce the interaction between their neighbor, they also reduce the interaction to the substrate. They become more isolated. Basically, after the transition, they stop listening to the community. They behave by themselves, which is a very important hallmark for the cancer metastasis. So recently, we have also performed the deep RNA sequencing analysis to analyze what gene might be activated in the R cells after the dissociation. What we find is, in the R cells, there are about 90 genes differentially expressed in comparison to the E cells. Among those 90 genes, many of them are related to the apoptosis inhibition and related to the cell migration promotion. In particular, there are two genes related to the cancer stem cells. So all these results suggest during this transition, the R cells not only show the phenotype change, but also show the genotype change. And noticeably, all this transition is due to the mechanical microenvironment stimulation. So as a summary, what we discovered so far is if the mechanical microenvironment is appropriate, the HCT8 the cancer cells can express a series cancer metastasis hallmarks, including the reduced cell-cell adhesion, increased the cell motility activity, the upregulated invading capability as verified by the invasion assay, the upregulated apoptosis inhibition gene, as well as the cancer stem cell genes. Also, we find that this transition is permanent, and it can be triggered by the appropriate mechanical signal, but it cannot be reverted by the mechanical signal alone. So all what I'm saying here is during the cancer metastasis, the onset could be triggered by the biochemical signals, but the mechanical signals may not be neglected, and they may play as important a role as the biochemical signals. Thank you very much, and I would like to take any questions you have.
Thank you.
Yeah.
So on the soft gels, did you image the E-cadherin at the junctions, and what do you see in terms of is that basically on
Yeah, actually
it wasn't clear.
Yeah, this picture showing the E-cadherin standing on the soft gel, 20 kPa. This picture showing the dissociated cells on the 20 kPa gels. So both of them are the cells on the soft gel, but at a different cell state. These two pictures showing the magnified view of those black box. This is on the soft gel. Yeah.
Okay, so what I'm wondering is whether or not you're seeing a decrease in the amount of cadherin at the junctions, or is the actin site, and also too, what does the actin cytoskeleton look like adjacent to those cadherin junctions on the soft?
Yeah, that's a very good question. I didn't show the actin structure here, but we did the actin cytoskeleton staining. What we find is on the soft gel, the actin trace fibers in the cell island is diffused. Cells mostly show the cortical actin around the cell periphery. The only actin fiber showing up is at the bottom of the cell colony, but at the top of the cell colony, they don't have the clear filaments. So which indicated the cell on the soft gel actually are experiencing less intracellular force. We didn't, if you are talking about the co-staining of the E-cadherin and actin, we have not done that. I did the, yeah.
You answered my question.
Okay. Yeah.
Shin, what a beautiful talk and such interesting results. This is not a question, it's a comment. A couple slides before this, you showed us some, I think, 8 cells up in the upper corner, and each of the, that one, no, what, next one, that one.
Okay.
Now, what's really interesting about this is 5 cells, sorry.
I pick up a 5 random.
Emerging recognition that we're getting in many requests for funding from the National Institutes of Health, that we've been thinking about cells and tissues as being homogeneous. We have liver cells, we have heart cells, we have cancer cells, but in fact, they're very heterogeneous within a tissue. And what this is showing me is that this heterogeneity is showing up under these culture conditions, which is very interesting. That's probably in this case, part of the transition. And it is recognized, as far as I know from my interactions with Dr. Bhargava, that this is also characteristic of tumorigenic tissue. So the point I'm making here is look for heterogeneity. That's sort of what people are now, that's the future.
Yeah.
It's a very good point.
Yeah.
Very good point.
Specifically for single
Yeah.
cell analysis.
Yeah.
Exactly. Yeah.
So the idea that
Thank you.
among a group of cells, actually, so from your subgroup, right, is there a smaller subset, 1 or 2 or 3 cells that are really the ones that are most metastatic?
Exactly. Yeah. We consider about this. Yeah. There might be some bad guys to trigger this process. Yeah. I mean
Related question. So wasn't it, I thought that one of the sort of the going hypothesis was that the cancer tumor, the matrix or the overall tumor might be stiff, but the cells are softer and hence they
Yeah.
escape the matrix, right? So here you're showing that locally the matrix could be softer and maybe it's the soft matrix that triggers the cells to escape. So which one is it? Is it the cell or the matrix?
I think there is a terminology difference. What I'm saying is softer here. I mean 1 kPa is softer. 20 kPa I mean intermediate softer. But this softer is in comparison to 3 gigapascal. If you compare these two and compare them to the physiological stiffness, this is quite stiff. Based on the literature, these cells are from the colon. The colon stiffness is about 1 to 2 kPa. But when the colon cancer cells develop a tumor, those tumor generate fibrosis. People, not 20, we don't know. For the fibrosis tumor, colon tumor, the stiffness is about 3 to 4 kPa. That's not a metastasis yet. There are no data showing how stiff the tumor is before the metastasis. So what we observed is the trend from the 1 kPa to 20 kPa. If you keep the cells in this stiffness, but not softer. But not softer, the cell show this cell state transition. But what we are right now working on is we want to really find out what's really the stiffness of the tumor before metastasis. There are no data showing that.
Okay. So your work was 21 kilopascal in the polystyrene.
21 actually we have a series of stiffness. 21 things is optimal.
So in 1 or 2 kilopascal, it doesn't happen.
It doesn't happen.
They form the tumor, but they don't.
That's the stiffness of the normal colon. Yeah.
So what he's doing now is actually getting the tumors from the patient from Provena, and then looking into those tumors' stiffness as well as the cells.
But are you considering looking at the stiffness of the cells also? Because
I did that also. Yeah. After if you measure the single cell stiffness, they are really softer than the original cancer cells. Yeah.
By how much?
By 2 to 3 times. So after they
that's a very nice comprehensive study if you can correlate all of that.
Yeah. So you have done all the FM studies.
That's fantastic.
I saw the time is only 10 minutes, so I didn't put that results.
Sure. All right.
Yeah.
What about something like an osteosarcoma where the matrix is rather stiff, or something like metastatic prostate cancer, where it metastases to bones and forms bony tumors, where the matrix is actually inherently stiffer than, say, a colon cancer?
Can you repeat the question? I just heard about you talk about the bones. I didn't hear the complete question.
Osteosarcomas, where you have a bony growth.
Osteosarcoma.
Osteosarcoma. What about like a metastatic osteosarcoma? Because the matrix is actually stiffer.
Yeah.
Or metastatic prostate cancer, which metastases to bone. So have you thought about the transition that would occur there?
Actually, for the colon cancer cells, they metastasize to the liver instead of the bone. I think the breast cancer cells metastasize to the bone preferentially. So the
Breast cancer will degrade bone.
degrade bone, yeah, as well. Right. I think bone stiffness is too stiffer for the colon cancer cells.
I'm wondering if you think that the mechanism is the same, though. Or do
Okay.
you think it's different?
Yeah, I'm not very clear about that. Yeah. But it's a very good question.
Good. Okay, well, thank you very much to all the 4 speakers.